Next topic is PNC of zoonosis, that is prevention and control of zoonosis. So there are 11 approaches, and these approaches you can remember with the mnemonic from your ECG pattern, that is PQRS and T. So there is QT interval in the ECG pattern, and for this QT interval there are so many drugs which cause the prolongation of the QT interval. So the mnemonic here is QT interval improvement. QT interval improvement. These 11 approaches for the prevention and control of zoonosis can be remembered with QT interval improvement. QT means quarantine and T means test and slaughter. So QT, quarantine, test and slaughter. Then from the word interval, I for immunization, N for environment, T for treatment, E for early diagnosis and epidemiological diagnosis, R for reservoir control, V for vector control, that further include environment control, chemical control, biological control and genetic control. Then this AL means health education. AL means health education. And improvement means genetic improvement. Genetic improvement, that mainly through the selective breeding. With the help of selective breeding, some breeds can be developed which are resistant to some diseases. For example, we'll discuss about trypanosoma resistant breed. So these are the 11 approaches for the prevention and control of zoonosis, and their mnemonic is the QT interval improvement. So we'll discuss each approach with the important MCQ point of view. So first is the quarantine. QT interval improvement. So first we'll see quarantine. The definition of quarantine is the restraint or restrictions placed on good lamp. What is good lamp? This good lamp is goods, lower animal, M for man and plant. So restrictions or restraint placed on good lamp which are suspected of being carrier or vehicle of the infection or exposed to infectious agent. The quarantine period, it is given for different species, and for cattle it is up to 90 days, for pig and sheep it is for 15 days, and for immigrant dogs in rabies free areas like New Zealand, UK and Hawaii, in these areas for the dogs which are coming from the different countries, there is long term quarantine period because these areas are already free from the rabies. This quarantine period depends on two factors. One is incubation period and one is time taken for the confirmation of infection. So quarantine period depends on incubation period and time taken for infection confirmation. The first international livestock quarantine, it was used by the USA in 1890, and for human being the international or this quarantine period was used by Republic of Venice, Italy in present day in 1374, and this quarantine was imposed on plague exposed travelers. So these two are from the livestock and human, first time in 1890 and this is in 1374. Because of this quarantine, in 1924, organization OIE, it was established to make uniform procedures for the veterinary quarantine. So these are the important MCQs related with the quarantine. The key point is the for the definition, this is the restraint placed on these and quarantine period, that is for cattle, pig, sheep and rabies free areas 90 days, it is 15 days and it depends on incubation period and time taken for infection confirmation. And first international that quarantine used for livestock in 1890 and for human, that is for plague travelers, in 1374. And to regulate this quarantine in animal species, OIE organization was established in 1924. Next is test and slaughter. So next T for test and slaughter. Before going for test and slaughter, we normally do surveillance, and that surveillance is through mass testing of the population. So first is surveillance through mass testing, then test and slaughter. But this policy of test and slaughter is not possible in India because of some religious issues. So in India, what we follow is test and segregation policy. Test and segregation. For example, in case of brucellosis, we test the animal. If it is found positive, then it is segregated from the healthy animal. Cattle is not slaughtered in India. So test and segregation policy. But outside India, because of this test and slaughter, they have eradicated tuberculosis, brucella and rinderpest like diseases. Means if they want to eradicate, then they need to use test and slaughter. But here we are using test and segregation. For bird population, this slaughter or segregation term is not used. They are using depopulation. For example, if there is outbreak of HPAI, that is avian influenza, then the whole population is culled, that is the term uses depopulation in case of birds. After quarantine, test and slaughter, next approach is immunization, which is also known as mass immunization or vaccination. With the help of this immunization, we can reduce the number of susceptibles. Once the number of susceptibles are reduced in a herd, then if number of susceptibles are reduced, it leads to augmentation or increase in the herd immunity. So mass vaccination, the current program, that is National Animal Disease Control Program for FMD and brucella. So cattle, buffalo, sheep, goat and pig, and in brucellosis control program there is 4 to 8 month female calf mass vaccination is going on. And already these well planned programs have met their role, that is National Rinderpest Eradication Program that led to eradication of rinderpest, and pulse polio program. These vaccinations, these are under trial, like aerosol vaccination against bat means brucella, anthrax and tularemia, aerosol vaccination. Then oral vaccination with the help of via water for or against fox rabies, and nebulization or atomization vaccination for some group of animals. So these techniques are under trial, or they are not getting much result, but they have tried. So mass vaccination program which is going on NADCP, and because of NREP, rinderpest was eradicated. Environmental hygiene. Environmental hygiene is the first public health measure which is to be applied on population. So these are the some examples where environmental hygiene maintenance will control and prevent the zoonotic diseases. First is protecting public water supplies. This public water supply, if it is protected, means it is not contaminated, then we can control the population health. Improving domestic environment of the human or animal. Sanitary control of the sites of animal slaughter. Then systematic ante-mortem, post-mortem examination of the animal slaughter. And environment health on the farm. There is one system for pig production, that is McLean County system. Following this system of pig production, swine production, we can increase litter size or increase pork quality. Then implementation of following farm hygiene practices like proper excreta treatment and disposal, then pest control and availability of the clean water. So with these environmental hygienes we can prevent and control the zoonotic diseases. T for treatment, and this treatment is mass treatment, or you can say population treatment. There are two examples where we can use coccidiostrate in case of poultry via drinking water, and we can use antihelminthics in ruminant via feed. So treatment is the mass treatment or population treatment. Next, E for early diagnosis. This early diagnosis is very important for immediate treatment and control measures. And this early diagnosis, this method is applied, or specifically it is used for chronic infectious diseases and non-infectious diseases. For example, these are the diseases and these are screening test or early detection test. For tuberculosis, tuberculin skin test, which is nothing but a type of type 4 delayed hypersensitivity test. For brucella, that is herd test, milk ring test and RBPT serum test. Mastitis, yes, California mastitis test, where somatic cell count is detected. Glander's Malin test, and for Salmonella pullorum or Salmonella agglutination test are used. So these are the diseases and these are the screening tests for the early diagnosis of chronic infectious diseases like tuberculosis and non-infectious disease like your mastitis. Next, E for epidemiological diagnosis. Here, diseases with their frequency of occurrence and time of occurrence, place and their pattern of occurrence. For the diseases, these points like their frequency, time, place, pattern, these are ascertained or quantified. Purpose of this epidemiological diagnosis is to determine the immediate and long-term needs for the purposeful action against diseases. And the important aspect of this epidemiological diagnosis is mass testing of the population. And two principal components of this epidemiological diagnosis is epidemiological intelligence and analysis. And the uses of this diagnosis is to develop appropriate approaches against emerging infectious diseases like hemolytic uremic syndrome in human, Ebola, Lyme diseases, and use of some molecular techniques in epidemiology like PCR, RFLP, DNA probes. So these are the applications to principal component. The purpose and frequency, time, place, pattern are quantified in epidemiological diagnosis. R and V. R means reservoir control and V for vector control. Reservoir control. First, what is a reservoir? The definition of reservoir is any pen, P-A-N. Pen means person, animals and non-living thing. So any pen in which the infectious agent like bacteria, viral or any agent which lives and multiply and can be transmitted to susceptible host, that is known as reservoir. And the most common techniques used against these reservoirs are trapping and poison baiting. Then vector control, V for vector control. It is of following 4 types, that is environment control, chemical control, biological control and genetic control or genetic manipulation. Vector control. There are 4 methods for vector control. First is the environmental control or environmental measures. These are the 3-4 some environmental measures like source reduction, filling and drainage, disposal of the waste and plant water management for the prevention and control of zoonosis. Then chemical control. Which chemicals? The chemicals are insecticide. Which insecticide? They may be contact poison and stomach poison and fumigants. Contact poison may be natural or synthetic like pyrethroid, mineral oil, rotenone and dairies. Synthetic may be organochlorine, phosphate, carbamate. And stomach poisons, 2 examples, sodium fluoride, Paris green, fumigant, HCN or sulfur dioxide. Then number 1 is insecticide, number 2 is use of oil films on water to control culex, that is filariasis in human. Use of polystyrene beads on water surface to control Anopheles stephensii. And in China there are some bed nets are available which are impregnated with insecticide deltamethrin at the rate of 15 mg per meter square to control the mosquitoes. Then biological control, use of some biological agents like fish, fungi, protozoa and bacteria. There are 3 fish species, Gambusia, Aplochilus, Pachylia. And fungi there is only one which is most commonly used, Coelomyces. Protozoas are 3, Nosema, Thelochenia and Vorticella. Bacteria only one, that is Bacillus, and 2 species, Thuringiensis and Sphaericus. And last is genetic manipulation of the vectors. Through which technology? Transgenic technology, means manipulation of their DNA. So these are 4 measures for control of the vector. Last 2 approaches are AL is the health education and improvement is genetic improvement. So health education purpose is to make the community aware, to make the farmers or pet owners client aware of the zoonotic diseases, and that can be achieved through mass media like TV, radio, newspaper and others. Genetic improvement is through the selective breeding where some disease resistant breed are developed, for example trypanosoma tolerant breed and Dama. 11 approaches for prevention and control of zoonosis. Then 5 planning steps for the prevention and control of zoonosis. These are: first, define the objective for zoonosis control. Then apply the approaches for control of zoonosis in animals and control of vehicle, and prevention and treatment of zoonosis in human being. And last is the establishment of the surveillance, and this establishment is through the establishment of diagnostic services and epidemiological intelligence services. So diagnostic services means clinical diagnosis, lab testing, and epidemiological intelligence services include collection of data, processing and analysis, and then prompt feedback is given. So this is the establishment of the surveillance.